What are organoids in pancreatic cancer research? Imagine tiny, living models of a patient's tumor that can grow in the lab. These are called organoids, and they are revolutionizing how scientists study pancreatic cancer. Organoids are three-dimensional clusters of cells grown from a patient's tumor tissue. They are created by taking a biopsy or surgical sample of the tumor and placing the cells in a special environment that mimics the natural surroundings of the pancreas. This allows the cells to self-organize into structures that look and behave like the original tumor. These miniature models are especially useful because they closely resemble the biology of pancreatic ductal adenocarcinoma, the most common type of pancreatic cancer. They keep the genetic makeup and diversity of the original tumor, which is important because tumors can vary greatly from person to person. This means researchers can study how different tumors grow, spread, and respond to treatments in a way that is very close to what happens in real patients. One of the biggest advantages of organoids is that they can be grown and used repeatedly. Scientists can freeze them for future studies or analyze their genetic information to understand what mutations are driving the cancer. This makes organoids a powerful tool for exploring the molecular pathways involved in pancreatic cancer development. In research, organoids are used to model the disease more accurately than traditional cell cultures. They help scientists understand how tumors grow and invade nearby tissues. They also serve as personalized testing platforms. Doctors can test different chemotherapy drugs or targeted therapies on a patient's organoids to see which treatments might work best before trying them in the patient. This approach is a step toward more precise, individualized medicine. Scientists also use organoids to find biomarkers, molecular signals that can predict how a tumor will respond to treatment. This could help doctors choose the most effective therapies and avoid unnecessary side effects. Additionally, by growing organoids alongside immune cells and stromal cells, researchers can study the complex interactions within the tumor environment. This is important because the tumor microenvironment influences how the cancer grows and how resistant it might be to treatments. While organoid technology is still developing, it offers exciting possibilities for future therapies and prevention strategies. It bridges the gap between simple cell cultures and animal models, providing a more accurate, patient-specific view of pancreatic cancer. This helps researchers better understand the disease and develop new targeted treatments. However, it is important to remember that organoids are a research tool and are used alongside existing clinical approaches. They are not yet a replacement for standard treatments but hold great promise for advancing personalized medicine and improving outcomes for those affected by pancreatic cancer. Always consult healthcare professionals for medical advice tailored to individual situations. As research continues, organoids may become an even more vital part of how we fight pancreatic cancer in the future.